Studies of the distribution of genetic variation across the continents combined with archaeological information have revealed some details of our ancestors' global expansion. The journey along the fringes of Asia and through the archipelagos of modern Indonesia to New Guinea and Australia was accomplished about 60,000 years ago. Getting to Australia required crossing several substantial bodies of water, suggesting that our ancestors were already using boats at that early stage. Modern humans arrived in Europe around 40,000 years ago and penetrated northern Asia, including Japan, some 10,000 years later. Like so many other leaders in this field, including Rebecca Cann and Svante Pääbo, Michael Hammer at the University of Arizona received his training in Alan Wilson's Berkeley lab. And though Hammer's initial interest was mice, the publication of Cann and Wilson's mtDNA study diverted him from rodents to the human past. He was among the first to realize that information from the Y chromosome would provide the crucial test of Cann and Wilson's overall hypothesis. But the Y proved reluctant at first to yield its secrets. One study, done in Wally Gilbert's lab, sequenced the same chunk of DNA drawn from multiple individuals, only to find the sequence identical in every instance. A laborious effort that yielded zero information about genetic interrelations. Hammer persisted, however, and eventually he and others turned the Y chromosome into an anthropological goldmine, whose payoff culminated in Underhill's landmark paper. A major vein in the Y chromosome mine has enriched our attempts to reconstruct the human colonization of the New World, a relatively late development. The identity of the oldest human settlement in the Americas remains contentious. A site in Clovis, New Mexico, is the traditional title holder, dating back some 11,200 years. But fans of a site in Monteverde, Chile, claim it to be at least 12,500 years old. It is also debated whether the first Amerindians crossed the land bridge across the Bering Strait during the last ice age or took a more southerly route in boats. What the genetic data make clear, however, is that the founding group was small, with only 2 major classes of Y chromosome sequences detected. There appear to have been just 2 distinct arrivals, each perhaps involving no more than a single family. Among Amerindians, mtDNA variation is much more extensive than Y chromosome variation, suggesting that there were more women than men in each founding group. Probably the more common of the 2 Y chromosome sequences represents the first arrival. The descendant population would then already have been established before the arrival of the second group, which included the ancestors of today's Navajo and Apache. The more common sequence also boasts another distinction: the presence, first noted in 2002, of a mutation that is rarely found elsewhere on the planet, giving further evidence of its bearers' precedence as pioneers. This mutation is calculated to be about 15,000 years old, not much older than the earliest known archaeological sites. Genetic analyses have permitted the reconstruction of more recent phases of prehistory as well. Hammer, for example, has shown that modern Japanese are a mix of the Jomon, ancient hunter-gatherers, currently represented by Japan's aboriginal Ainu population, and relatively recent immigrants, the Yayoi, who arrived about 2,500 years ago from the Korean peninsula, bringing with them weaving, metalworking, and rice-based agriculture. In Europe, too, we see evidence of waves of migration, often associated with advances in agricultural technology. Groups like the Basques, who live in the mountainous Pyrenees on the French-Spanish border, and the Celts, who arrived later and are found throughout the northwest margin of Europe, from Brittany and France through Ireland and western Britain, are genetically distinct from the rest of Europe. One explanation is that each of these groups was displaced to relatively far-flung regions by more recent arrivals. Brian Sykes at Oxford has done much to reveal the complexity of the genetic map of modern Europe. Conventional wisdom had held that modern Europeans were largely derived from the Middle Eastern populations that invented agriculture in the Fertile Crescent between the Mediterranean and the Persian Gulf. Sykes, however, has found that most European ancestry can be traced not to the Fertile Crescent, but to older indigenous lines predating the incursions of Middle Easterners and to migrant groups from Central Eurasia. Such groups, including the Celts and the Huns, who swept into Europe from the east around 500 BC and AD 400, respectively. And taking his analysis of mtDNA a step further, Sykes has argued as well that virtually all Europeans are descended from one of 7 daughters of Eve, his term for the surprisingly few major ancestral nodes in the European mtDNA family tree. A company he founded called Oxford Ancestors will, for a fee, sequence part of your mtDNA to determine from which of the 7 daughters you are descended. Another key to understanding the human past may rest with an observation fruitfully exploited by Cavalli Sforza and others. Patterns of genetic evolution often correlate with those of linguistic evolution. There are, of course, the obvious parallels between genes and words. Both are transmitted from one generation to the next. Both undergo change, which in the case of language can be particularly fast, as any parent of a teenager knows. Likewise, American English is similar to, but distinct from, British English, even though the two have been evolving separately for only a few hundred years. On the basis of the similarities and differences, then, the family tree of languages can be reconstructed in much the same way the genetic family tree can. But even more important, in many cases, as Darwin himself first predicted, we can identify instructive correspondences between the 2 trees, such that what we learn about the one can deepen our understanding of the other. In The Origin of Species, Darwin notes, "If we possessed a perfect pedigree of mankind, a genealogical arrangement of the races of man would afford the best classification of the various languages now spoken throughout the world." Both the Celts and the Basques offer dramatic cases in point. Each people is genetically isolated from the rest of Europe, and each one's languages are correspondingly distinct from those of the rest of the continent. As for the New World, a controversial linguistic theory proposes that there are but 3 major language groups native to the Americas, and 2 of these correlate with the 2 early immigration events discerned in the Amerindian Y chromosome data. The third, by far the smallest, involves the isolated Inuit.